Le projet de présentation sera donné par Thomas Brown. Thomas est un candidat au PhD dans le laboratoire de Michel Caillouette. So our next speaker will be Thomas Brown, who is a PhD candidate in the laboratory of Michel Caillouette. And he's going to tell us today about connectomics. Thank you, Thomas.
Hello, everyone. My name's Thomas, and I have a confession to make. Despite being a PhD student in neuroscience, I don't actually have an undergraduate degree in neuroscience or biology. My undergraduate degree was in archaeology. And for those of you that don't know what archaeology is the study of items, very, very old items, and how those items tell a story of humanity. One of those things, and one thing we got very excited about when there was a discovery, are these. This is a map. It is the oldest map in existence. It was discovered at the site of Babylon in modern-day Iraq. And maps are extremely important because not only do they tell us where we are, they tell us who we think we are. For example, this little dot here is the city of Babylon. And all of these circles around are the countries and the cities which surrounded it, pushed far to the extent of the world around them, because people here would grow up and they would die in the city or a matter of miles away from it. This is a mappa mundi. It is one of many examples during the medieval ages of maps. And one of the main points of these maps is the countries that the people were from are grossly overproportioned compared to the rest of the world. Even when you open up your cell phone to find out where you are in space, you are at the center of the world. During my archaeology degree, I took a job caring for elderly people. For 3 years, these people became my friends. In some cases, my best friends, because I saw them a lot. And unfortunately, firsthand, I was able to see how diseases of the central nervous system, the brain, the spinal cord, and the retina, slowly stole from them their lives. It took away the lives of their families, and it affected the life of their friends as well, like myself. It was this that made me want to become a neuroscientist, to be able to contribute somewhat to the understanding of not only what makes you, you, but also somehow contribute towards understanding these diseases for which there is no cure. And I can tell you, and many of you probably already know, it is very sad to see the slow decline. This is a human brain. In my opinion, it is the most beautiful thing in existence. But I'm a little biased. Everybody has one, well, except maybe me. And already in this MRI scan, you can appreciate the complexity. So many of the areas look so different to one another. And this has been a question for millennia. What do these individual areas do? And how do they relate to one another? What are the connections? The idea of mapping the brain, mapping the mind is not novel. It's been going on for thousands of years. One of the main issues we have is the scale of the map and the size of the brain. The brain, as some of you may know, is made up of individual cells called neurons. We'll call them brain cells. And in your head right now are 86 billion neurons, brain cells. Each one of these can connect to potentially thousands of other cells. The estimated number of connections in your brain right now is larger than the number of stars in the Milky Way. In fact, it's a lot larger. It's 1,250 times the size of numbers in the Milky Way. That number is absolutely staggering. So why am I standing before you right now saying, oh yeah, let's draw a map of it, no problem. let me give you an example. I've just come up with a great business idea and it's going to make us millions. We're going to meet and we're going to talk about it, okay? And I want you to meet me at the coffee house. I'm not going to tell you which one. I'm not going to tell you where it is. I'm not going to tell you what time. And I'm not really going to mention what we're going to talk about. Do you think that's a reasonable request? I don't think it's a reasonable request. So why, when we talk about regenerating the brain and transplantation, do we think that we can just plant a neuron into the brain and it's going to find its way when we don't even know where it's supposed to go? There are many different scales of maps, as there are many different scales of connectomics, or the maps of the brain. This is an electron microscope picture right here. This is about 1/100th of a millimeter in size. It's tiny. And what scientists are doing is they're actually taking tiny thin slices of brain tissue, taking a picture, taking another slice, taking a picture, guess what? Taking another slice, taking a picture, and they're doing this millions of times. Now, as you can appreciate, the detail of these pictures when they're recreated in 3D is incredible. It's absolutely beautiful to see. The problem, 1 millimeter squared of this tissue took researchers 5 months of non-stop electron microscopy working. As you can imagine, that's very expensive and it's very difficult. So we can make the scale go up. We can have a bigger scale. This is an image that I took. It's basically some cells in the retina that I've infected with a virus just to make them glow green, right? And the reason I did that is because we can actually take a 3D image of this and we can reconstruct it by using the microscope. Now that's really, really useful. And I think it's pretty cool as well. Now, there's another scale that many of you have probably heard of, and some of you might even have had. So this is an example of a very special MRI. It's a special technique which uses MRI, and it actually shows you the tracts of your nerves. It can show you the nerves in your brain. And further than that, it can actually show you what they're doing, what they're firing, what they're communicating with. Now, the issue with this is it's pretty expensive. So scientists like myself use model organisms. So that basically just means we get an organism, like a mouse in my instance, and that means we can do certain manipulations, and we can visualize this on a lot smaller scale and try things that are new. So this is a technique that I use. It's called brain clearing, and it allows us to make the brain of mice completely transparent after injecting a tracer. So that's just something which allows us to visualize, you know, the neurons and the nerves in the eye. So this process is pretty cheap and it renders them completely see-through, and it allows us to scan these brains in 3D. So I just have some for you if you want to take a little look. Please don't break them. I need them for my project. So we have a before and an after, and there's just a video here as well. So could you play the video, please? And the brains that you're looking at right now, after I scanned them, I could see beautiful, beautiful kind of tracks and connections. And this is really interesting because it allows us to kind of ask interesting questions like, what changes during development? How do these connections form? Not just what are these connections? One of my favorite things about connectomics, the science of connections, is not just that it allows me to kind of visualize one of my favorite things, but also because no one person can map an entire brain. No one person can do this. So naturally, by its very nature, this is a collaborative science. And this means that all of the little islands that PhD students find ourselves in and professors find themselves in, just studying kind of and not really collaborating as much and feeling a little lost, it kind of goes away. So not just are we learning about connections between in the brain, we're also connecting with each other. And there's many different questions you can ask based around the scale. And that's why I think it's incredible. And I hope now you're at least a little more excited about this than you were before. And I hope that I've told you how we are trying to learn more about ourselves. And hopefully one day we can help some people along the way. Thank you.
Thanks, Thomas. Thanks for bringing some brains too. That's pretty cool.
Yeah, please don't break them. That's not a joke.